Hello and welcome to BIOS Frontier Science. I'm Kate Shannon, and today I'm excited to welcome César de la Fuente, President Assistant Professor at University of Pennsylvania. César, to kick things off, could you please give us a quick introduction to yourself and what you're working on?
Yeah, sure. Well, first of all, it's really a pleasure to be here. My name is César de la Fuente. I'm a Presidential Assistant Professor at the University of Pennsylvania. And what really excites me and excites us in my lab is to use technology at the intersection between machines and biological systems to be able to better understand, predict, and treat infectious diseases. So that's kind of what gets us all out of bed every morning and what really drives us every single day.
That's awesome. And so, like you said, that seems to be the common theme in your work. Could you expand on that a little bit? Because your lab seems to be working on several distinct areas, but what are the common threads that tie together?
Yeah, absolutely. So we're a highly interdisciplinary lab. So we use principles from different disciplines, including biology, microbiology, computer science, synthetic biology, and so on. And so one of the main things that we're trying to do is we're trying to develop novel antibiotics. And as you probably know, the cost for drug discovery and drug development has essentially steadily increased over the last 6 decades or so. And nowadays, to take a drug from the laboratory to the clinic, it costs over $1 billion. Just to put this into perspective, this is more than the budget that NASA has to take a rocket to the moon. And so it's incredibly costly to discover novel drugs and to actually take them to the real world where they can have an impact on patients and on people. And what we're trying, and not only is it a highly costly process, but it also takes a long time. So on average, again, to develop a new drug, it takes around 10 years. And so you have to wait a long time to see something have an impact in the world. And what we're seeing now is a new phenomenon, as you know, with the increase in compute power following Moore's law, with the ever increasing number of transistors that we can get into a chip. And that has really enabled this computational revolution that I think can have a huge impact in drug discovery. So of course, by using computers, we can process vast amounts of data, and we can also try to shorten the time scale that it takes to again discover new drugs and to develop them into something tangible. And that's what we're trying to do in my lab. We're incorporating principles from AI to help us do that, and we're hoping to have an impact in the field or the space of antibiotics. The reason we care about antibiotics is that they're really essential tools for modern medicine. So modern medicine would not exist with antibiotics. We need them not only to treat infections if we get infected, but also for childbirth procedures, for chemotherapy, for any kind of surgery that you can imagine. You need antibiotics to sterilize the area, and so without them, we would be pretty much lost in terms of our ability to perform contemporary medicine.
Yeah, awesome. So, like you mentioned, part of your lab is working on the development of novel antibiotics. Before we dive into the details of that project, could you give us an idea of why it's important to start developing new antibiotics?
Well, in addition to what I mentioned, that they're really essential tools for medicine, the current projection at the current rate is that by the year 2050, around 10 million people will die from drug-resistant infections. These are bacterial infections or bacterial cells that have developed resistance to available antibiotics that we have today in the hospital or in the pharmacy. And if you do the math, those 10 million deaths per year correspond to approximately one death every 3 seconds. And so that's the world that we're heading towards. It's a post-antibiotic world where these drugs no longer work. And I think here it's important to emphasize that there have been 3 main pillars that have allowed humans to practically double our lifespan. One of them has been vaccines, which we have seen an incredible renaissance now with the mRNA technology. We can talk more about that. Clean water, drinkable water, and then the third pillar is antibiotics. So if we suddenly take out that pillar from the equation, we're going to be in a world where even a minor scratch, from falling while playing on the playground can lead to an infection, can lead to sepsis, and can be a situation of lethality where the individual can die from a simple infection. So it's incredibly crucial that we give more visibility to this global health and public health problem that affects not only people here in the United States, but people everywhere in the world. This is agnostic to where you come from. It affects everyone. It actually affects more individuals. It affects more in a more pronounced fashion individuals that come from low research settings and developing countries. And so obviously that's one of our motivators as well, to try to help people that need it most. And that's something that we always keep in mind. I always tell the team here in the lab that to have this sense of urgency, right? We're actually trying to develop these new drugs for patients that need it today. And I actually, I get letters from families and patients that have daughters and sons and in the hospital with incurable infections, and that's a great reminder of the urgency that this topic imposes, and basically just that's really what helps us get out of bed every morning, actually.
Yeah, absolutely. There's a need for these antibiotics now, and it's only going to get worse over the coming years. And so your lab is working on two approaches to develop new antibiotics, synthetic peptides and looking for existing antibiotics in nature. Could you give us an overview of these two approaches?
Yeah, for sure. So yeah, one of the things that we try to do is we try to find antibiotics in nature. So we do bioprospection expeditions, mostly computationally, to try to find new antibiotics in genomes and proteomes of either contemporary organisms, but we're also doing molecular de-extinction projects where we're trying to de-extinct molecules from extinct organisms. That includes the woolly mammoth, it includes ancient humans. And so we're open to really exploring all sorts of information, be it DNA or RNA or amino acid information from genomes and proteomes, to try to find entirely new molecules that perhaps today in our society can have an impact. And so that's the natural side of things in the lab, where we're trying to find molecules in natural data sets and natural data. The other aspect that we do is we're trying to computationally design synthetic molecules, well, computationally and chemically, and biologically, actually. But through experimental design, what we do is we either tweak naturally occurring sequences of different molecules. Mostly we focus on small proteins. The reason for this is that proteins mediate all biological processes that we can think of, including capturing energy from the sun, even thinking or moving. Really, any sort of reaction that allows us to exist is carried out by proteins, and small. In our case, we focus on small proteins called peptides. But what we do is we can synthetically, either in the computer or through chemistry, create novel molecules, either by using natural ones as inspiration, or we can de novo also create entirely new molecules. And we focus on antibiotics. So I can tell you about one of the projects that we published relatively recently, where what we did is we encoded small peptides from nature onto the computer. So again, we basically translating that information from those chemistries into ones and zeros, so the computer can comprehend that molecular structure. And then we thought for a while, what's the best way to innovate at the molecular level, or at any level, really. And we came to the conclusion that the best way to innovate and to create diversity was to actually follow the same phenomenon that has allowed all of us to be where we are today, right? Throughout the evolutionary process. That's really the best way to generate diversity. And so what we decided to do is we decided to train the computer to execute Darwin's algorithm of evolution. So the theory of natural selection, but in this case, it turns into the theory of artificial selection because we're doing it on the computer. And so the time scale that it takes, we really compress the time scale that it takes to evolve something. So instead of having to wait for millions of years for a molecule to get evolved, we can do that in really in days to weeks on the computer. And the computer was successful in doing so, was able to evolve the molecules to create novel synthetic ones that it predicted would be good antibiotics. And of course, at this stage, that's only a prediction. But then in my lab, we not only have the computational design aspect, but we also have an experimental branch where we can validate everything that the computer is predicting. And we were able to synthesize those molecules that the computer generated. And we showed that some of them were really good at killing bacteria, not only in vitro, in Petri dishes, experimentally, but also in a number of animal models. And so that's really what sparked this new field of trying to design novel antibiotics using principles from AI and computer science. And really, the dream now is to answer the question, can we design an AI generated antibiotic that actually and take it to clinical trials? That's really the dream that we have at the moment.
Interesting. And we'll get to the validation aspect of it in a little bit. But diving into the AI side of things, what are the major challenges of an in silico approach?
I would say the main challenge in biology and medicine as a whole is that we rely on data sets that are highly heterogeneous. And we rely on data sets that have not been generated in one place using standardized conditions. And that makes it makes the reliability very complicated. And so one of the big efforts that we put a lot of our attention on early on was actually to develop our own data set in the laboratory using standardized methods, the same medium, the same bacteria that we have characterized thoroughly, and having all those standards in place to be able to produce a data set that is highly reliable and robust. And that's what we've been doing for the last couple of years. And I think really that's going to be one of the keys for any drug discovery AI effort, not only in the antibiotic space, but also in other kinds of drugs. You can think of anti-cancer drugs or drugs against other infectious diseases like malaria or even COVID-19. And I think data generation is one of the key things that will play a huge role in the future.
Interesting. So my next question was actually going to be, what data do you guys use to train and develop these algorithms? But I've already answered that one. So I guess what sets your data apart from what other people are using or other approaches that are out there?
I mean, I really think it's the fact that we've devoted a lot of our time and effort and resources in generating something in-house. So every process of the way of generating data, all the metadata that is created around the data set that we have, we understand and we control every step of the way. And I think that allows us to, if whatever prediction we get, it doesn't make sense for some reason, it allows us to go back and reverse engineer the problem. And I think that makes it a little bit less of a black box, the whole process. Although, of course, if you're using machine learning and deep learning models, there is also a huge component of the black box notion that you don't know what's going on in between. But at least on the chemistry side and on the data generation side, we can reverse engineer the problem and go back and ask questions about each step of the way. I think that's a huge that's a huge resource that we've been able to generate.
Yeah, no, it's amazing. And so once you have these algorithms, how do you validate them and make sure that they're working the way you expect them to? Is this something that you're going to have to wait till you're actually trying these antibiotics in humans, or are there kind of earlier milestones that you expect to hit that will, validate these computer models?
I mean, the way we're doing it at the moment, and that's a great question. I think validation is absolutely essential. And that's why, again, our lab is composed of both the computational side and the experimental side, so we can validate absolutely everything the computer thinks, that this might be a good antibiotic or this might be a good drug for this indication. We validate everything in the lab. And so the way we're doing it is we're validating everything first in vitro. We have a number of assays that we've developed and streamlined, and that allows us to answer questions like, the easy question or the most obvious question: is this new molecule capable of killing gram-negative bacteria or gram-positive bacteria? We're of course targeting the main pathogens that endanger our society, including, Acinetobacter baumannii, which is also so-called the Iraqi bug or microbe that typically infects soldiers in the battlefield. We also have MRSA, methicillin-resistant Staph aureus, which causes a lot of problems in hospitals and highly recalcitrant infections. We target Pseudomonas aeruginosa, which is a gram-negative bacterium that affects, for example, patients with cystic fibrosis. It infiltrates their lungs and causes these very exacerbated infections that are highly difficult to treat, and they're chronic. And anyway, they really worsen the condition. And so we validate everything in vitro, and then we have a number of biophysical assays that we run also to ask questions such as: are the molecules targeting the outer membrane in the case of gram-negative bacteria? Are they targeting the cytoplasmic membrane? How is that working? Are they just poking holes in the membrane and killing the microbe, or are they doing something else? Are they going in? Are they interfering with metabolism? Are they binding to DNA? How exactly are they killing the microbe? And there are really a number of ways that you can kill a microbe. And so we try to poke on each of those to try to gain as much understanding as possible. And then we've developed also resistance development assays where we ask the question of are these do bacteria develop resistance to these new synthetic molecules that we're exposing them to? Of course, one of the things that we don't want is for bacteria to develop resistance very rapidly, right? And so we also worry about that, and we have assays to probe that. And then we've developed a number of in vivo preclinical mouse infection models in the case, to evaluate the antibiotics that we're designing. And we have 4 or 5 right now that we've streamlined as well. And so we really validate everything experimentally at different levels.
Makes sense. And so kind of taking a step back and looking at AI for drug design a little broader faculty, are there any significant differences in using AI to design or discover antibiotics versus, other types of therapeutics for other diseases?
I mean, I do think that's a good question. I do think that a lot of at least some of the elements that we're developing and working on, I think you can extrapolate them to other indications or other fields even. So for instance, the genetic algorithm, right, that we developed to evolve the antibiotic, you could use that to evolve some other sort of molecule or even a protein or a peptide to target, specific cancer cell. You could think of that, of framing that question like that, right? But I think in each case you need to tweak the tools that we're developing to you need to tailor them for the specific question or problem that you're trying to tackle. We've done a little bit of work with trying to target cancer cells while leaving unaffected healthy cells. We've done also a bit of work trying to build molecular tools to reprogram microbiomes. So the good beneficial microbes that are all in and on our bodies, most prominently the gut microbiome. So these are the really millions of microbes that live in our guts that influence many aspects of our physiology, including enabling digesting our food, and they really impact many ways of our health and or propensity to disease. And so we're also building tools for that. So again, that's kind of what we've had time to do, but you can potentially extrapolate some of these AI tools to other many different problems.
Cool. And we'll get to the microbiome and living medicines in a little bit. But one more question on kind of the AI stuff, or AI for drug development. So you guys are developing small peptides rather than full proteins. So when you're using AI to design peptides, how do you balance the trade-offs between the increased versatility that comes with, larger size, with the increased modeling complexity that comes with the larger peptides? And have you found like a sweet spot in this process?
Yeah, I mean, we prefer smaller peptides, and I'll give you some reasons for that. So, their small size allows you to synthesize them chemically. So with a simple chemical robot, like a solid phase synthesis robot, you can synthesize them in a laboratory, and then you can easily test them. So you don't rely exclusively on recombinant production, where you have to encode the particular peptide of choice, or typically larger proteins, onto DNA, the genome of a microbe like an E. coli cell or a yeast cell, to be able to use these microbes as factories for the production of these molecules. So with peptides, the interesting thing is that you can do both. You can do chemical synthesis, and you can also biologically produce them in different microbes. The other thing that you can do with smaller sequences is that you can explore them using machine learning and AI more easily as well. And I would say those are probably the two primary advantages. We have worked in the past with some proteins as well, and in fact, we have done an exploration using a pattern recognition algorithm to explore the human proteome as a source of what we call encrypted antibiotics. So these are antibiotics that are really hidden within larger proteins, and that upon cleavage with a protease, they get released, and then they exert their antibiotic properties. And in that case, we did explore a number of proteins. But again, I do think there's that advantage of the smaller peptide that gets cleaved, that then it's still highly bioactive. That's the other thing. And so that's what we prioritize, and we prefer to work with smaller sequences.
Makes sense. When you say smaller sequences, what size range are you talking about?
Typically, we work with between 5 and 45 amino acids, but probably the bulk of the molecules that we've generated so far is between 5 and 20 amino acids, so 25. Great. Yeah, and these are typically linear sequences, so chains of amino acids, one next to another, kind of like a collar of pearls, and each pearl representing an amino acid. And yeah, that's kind of what we've done so far, but the bulk of what we've done. But we've also played with cyclic structures and disulfide bonded structures and more complicated chemistries.
Interesting. And so you mentioned the microbiome a little earlier. The microbiome right now is emerging as a promising therapeutic target, and this is another place where your lab is spending a lot of time. Could you give us an overview of how the microbiome impacts diseases?
Absolutely. So the microbiome, in particular, the human gut microbiome, but let's not forget that we also have the oral cavity microbiome and the skin microbiome that is really all throughout our skin, which is the barrier that separates us from the outside world. But the emphasis in the research community has been on the gut microbiome, and for now probably 15 years or more, leading scientists have identified correlations of gut microbiome bacteria and a wide plethora of diseases, from susceptibility to infection, to cancer, to neurodegenerative diseases. I mean, you name it, practically any disease that you can think of, these microbes have been linked to those diseases in one way or another. But one of the things that I feel the field has really been limited by is the lack of tools to be able to reprogram those communities to be able to establish causal relationships. And for that, you need something like an analogy would be like in the field of genetics, how you have gene knockouts, where you knock out a gene to be able to understand its structure and function. Basically, that's how we're trying to think about this, and we're trying to develop molecular tools that allow us to knock out specific members, specific bacteria within a community of microbes, within a consortia of microbes, to be able to understand what that particular microbe is doing and how it's influencing disease, whether it's just a correlational relationship or whether it's a causal relationship. And of course, that's a very important distinction. And yeah, so that's what we're working on. We're developing such tools so that we can ask those questions in the future. We can say, let's say if you have, hypothetically, 3 bacteria involved in disease X, you have bacteria A, B, and C. If you have a molecule that targets A but not B and C, you'll be able to answer the question of whether A was causally influencing that particular disease X. And so that's kind of the concept behind this technology development efforts that we're doing in the lab.
That makes sense. Can you give us a few more details on actually how you're developing, or what tools you're developing that actually select for these bacterial populations?
Yeah, sure. So we're playing around with small proteins, and we've been basically able to design small proteins that selectively target individual microbes within microbial communities. This includes pathogenic microbes, but also includes members of the human gut microbiome, the skin microbiome, etc. And so we're basically we're tweaking these molecules chemically to be able to really exploit that selectivity to levels, to even the species levels, to be able to discriminate between species, to be able to just knock out specific, really specific members that we want within a community almost on demand. And we're developing really like a catalog of these molecules so people can choose, if you have bacteria, X and Y, and you want to target both, you can also do that. Or if you want to target just one or the other, you can also do that. So we're trying to really build a catalog that allows us to really reprogram and remodel these communities almost on demand.
Makes sense. And so you guys are right now in the stage where you're doing this to look for causal effects of, what populations you have in your gut, what health impacts does that have? And so what do you think are the most promising applications for this type of research, and what diseases do you think microbiome engineering can be used to treat in the future?
I mean, I think the possibilities are endless, really. Hypothetically or conceptually, really any disease that has been associated with a microbe or a group of microbes would be susceptible to being used within the context of this technology that we're developing. And so I'm very excited at the possibilities. I mean, I'm excited about learning about all these different diseases. I have, here at the University of Pennsylvania, excellent colleagues in, for example, psychiatric disorders who can start to probe whether specific microbes are playing a role in depression or in anxiety disorders or in even neurodegenerative diseases such as Parkinson's or Alzheimer's. We can start thinking at, start probing the role of these microbes in different types of cancers. Really, all the most devastating diseases that we can think of. I think we have a chance here, and I'm just excited at the possibility of continuing the research along those lines.
So what do you think are the most, prevalent challenges in designing or pushing this field of microbiome engineering forward?
I mean, in the context of using synthetic molecules, it will probably be getting FDA approval to be able to use them to, anytime you need to introduce something new into the body that is not natural. Obviously, there are a number of regulatory hurdles that have been put in place. And so we just need to go through the motions of that. Of course, making sure it's safe, not only efficacious but also safe. It's a key milestone as well. And that is if you need to put inside the body. If it's something where you can take a microbial community ex vivo outside of the body and then reprogram it and then reintroduce it, and that also comes with a number of hurdles, but probably not as many as when you introduce the molecule inside the body. So those are things that we're thinking about at the moment. And again, we're excited to hopefully get to a point where we get to conversations with the FDA in the future, and actually start thinking about trying to help people's lives and people's livelihood.
And as you start to overcome these challenges and actually start pushing some of these therapeutics into the clinic, where do you see this field going in the next 10 to 15 years?
I mean, my hope is that the microbiome engineering field will intersect with what we know as personalized medicine, where really the microbial composition that each of us has is highly diverse and different. And so I'm hoping that in the future we all have really a scan of our microbiomes, including the gut or the skin and the oral microbiome, and we know the composition of really our little friends that are that have co-evolved with us, that live inside and on us, and we understand how they relate to us, how they help us, and then how they can be harmful to us. And then hopefully we have drugs that allow us or interventions or approaches that allow us to remodel those communities really almost in real time and on demand, to be able to reverse certain disease conditions or even revert others. So that's really my hope. I mean, I guess thinking within the framework of personalized medicine, and then where for each individual, perhaps you can have a drug, or each groups of individuals that you can cluster, depending on their microbial communities. You can drugs for those individuals. So something like that, where we really take advantage of increased knowledge of those microbes that are again that are inside of us, and then create drugs that are tailored towards those compositions.
Yeah, that's amazing. It'll be fun to watch and see what happens over the next couple of years. So moving on to another project that your lab is working on, living medicines. Could you give us an overview of living medicines and the advantages that they have over traditional therapeutics? Probably first just start by explaining what are living medicines.
Sure. Living medicines are microbes that you can endow with the ability to produce any kind of pharmacological drug or medicine. So in the case of proteins and peptides, of course, you can, because they're composed of amino acids, you can encode that into DNA. So you can take the genome of any microbe, let's say E. coli, which is a gut microbiome bacteria, and you can encode it there, and then from the genome itself, the bacterium, as it's growing, it's replicating and it's producing the molecule of interest endogenously, and then you can basically teach it or train it to secrete the molecule outside of the cell, and then you can easily collect that particular drug, and then you can purify it and use it as a regular drug. But in the case of the concept of living medicines, so some of the things that we've done is we've taken, for example, microbes such as the ones that we have in yogurt, right? We have millions of probiotic bacteria in yogurt. You can engineer them in the laboratory to endow them with the ability, again, to produce any kind of therapeutic you want. In our case, we focus on peptide therapeutics. What you can do then is you can take the yogurt with your engineered microbe, it goes through your stomach, it reaches your gut, and once it gets there, it starts colonizing and replicating. Those probiotic bacteria start replicating, and as they replicate, they start producing this therapeutic molecule, and they start secreting it to the gut microenvironment, And let's say they have a salmonella infection. If you have a therapeutic that kills salmonella, then you may be able to resolve that infection. So that's the idea of living medicines, is really utilizing living cells as the medicine themselves, where they act as factories that produce, a given drug that allows you to resolve a particular, infection or disease. That's the concept underlying living medicines.
And what advantage does a living medicine have over kind of the more traditional approach that you explained?
I would say that it's a treatment in real time. Other things you can do, at least in the laboratory, you can induce expression. At least, when you use mouse models, you can induce expression by using what we call inducers, things like different sugars, like arabinose, for example. And so if you feed the mice arabinose, that can induce gene expression for the particular therapeutic that you're interested in. And so simply by feeding mice arabinose, a particular sugar, you're inducing the expression of the therapeutic almost on demand. So you can do things like that. You can also repress it. So you can really modulate the amount of drug that you're producing inside of the body really in real time. And I think that's a big advantage over traditional therapeutics, where you need to take them exogenously, and it takes a lot longer.
Makes sense. And so you mentioned inducible promoter systems, but what are some other key components to enable this technology? And are there any specific components that your lab is working on?
Yeah. So in addition to inducible promoters, you have secretion systems. So the inducible promoters promote the expression of the gene that encodes for the therapeutic, but that is only produced inside the cell, right? Then you need to get that outside of the cell to have any sort of effect. And for that, you need to think of different secretion systems. And really, that's where synthetic biology comes into play. There are a number of mechanisms that you can use to incorporate that into your particular problem. We've used in the past, for example, yeast cells. The reason for that is that yeast cells are really good at secreting molecules from inside the cell to the outside world, and they're very effective. So the secretion rates are highly, very, very high. But you can think of other things like secretion systems and other ways of basically pumping out things from inside the cell to outside the cell. Yep.
And so there are a couple different companies pursuing this general idea, but there's a lot of different cell types that are being used, from E. coli to mesenchymal stem cells to T cells. What do you think are the best cell types, and why is your lab focusing kind of on a gut microbiome, I assume E. coli?
Yeah, we're mostly focusing on microbes. I mean, the reason for that is just our expertise is really in that arena at the moment. We've used E. coli Nisl 1917, which is one of the prototypical probiotics that has shown efficacy as a probiotic in humans. We've also used yeast cell, like I mentioned, yeast cells like Pichia pastoris, which is again highly used as well for production and secretion of different therapeutic proteins. We haven't done as much work with eukaryotic cells, but those are also excellent options depending on the kind of problem that you're working on. In our case, because we're interested in infectious diseases, the concept here would be to engineer a beneficial microbe that really you can introduce into the body in a safe manner, and then endow it with the ability to produce a therapeutic to target a particular infectious disease. And so that's kind of what our focus has been. We have done a bit of work in cancer, but again, it's still really ongoing, and I don't have a lot to report quite yet.
And so, infectious diseases, do you have plans like kind of merging some of the projects and going on in your lab with the antibiotic design in this living medicine?
Yeah, absolutely. So we've done work where we've generated living medicines that produce antibiotics, and we've shown that you can target the bacterium of interest in a realistic in a mouse, in a real host in a mouse. So that's already at that stage, and we're really excited about the future. Of course, for living medicines to become a reality, we, society and the FDA have, it's going to take some time basically because it's really an engineered system. Although we do take probiotics every single day. We literally every time we drink kefir or eat yogurt, you're taking in millions of microbes. The idea there is that they're natural microbes, but once you introduce an engineered organism, that all obviously has a lot of ethical connotations and regulatory connotations, and so we basically need to go through the motions and the process of the FDA and other agencies.
Interesting. So my next question was going to be the biggest challenges, but you just answered that.
Yeah.
So when I was going to ask, where will we be in 10 years? But it seems like for this kind of specific branch, the more prudent question would be, how long do you think we start, or how long before we actually start using these types of living medicines? How long before they enter the clinic and, actually get to the commercial stage?
I mean, I think it's going to probably take some time. It's difficult to predict the future. The future sometimes comes earlier than we thought, and sometimes it takes longer than we thought. So it's tough to say. I would imagine within the next 10 years, we'll be able to make a lot of progress for sure. And I think society will catch up and regulatory agencies will catch up at that point. I do imagine a future where living medicines are able to detect disease also, not only treat it, but also in addition to treating it, detecting disease or diagnosing it, and again in real time. That's an interesting thing, and they can report on that information almost in real time. And I hope that within the next 10 years we see a lot of progress in that arena, and with the overarching goal of really being able to help patients and people, and yeah, hopefully we the community, scientific community, continues to be creative and continues to push forward to really take this to the next level.
Oh, it's very interesting stuff, and hopefully we see the technology develop a little further. But we spent most of this conversation talking about things that are probably a ways out before they actually get into humans.
Right.
But your lab is also working on a few technologies that have much more current applications. And one of them is low-cost diagnostics, specifically low-cost diagnostics for COVID. Could you tell us a little bit more about the test your lab developed?
Yeah, sure. So when the pandemic hit, really, we were working on a number of diagnostic technologies, mostly for bacterial infections. But with the terrible pandemic, really, at the very onset, I sat down with my team, and I really felt this sense of responsibility. As a researcher in the infectious disease field, working on technology development, I felt this responsibility to contribute something, right, to this pandemic that we're all going through. And so I sat down with the team, and we came up with a number of ideas to develop low-cost rapid diagnostics. And we really decided that the cost and the speed of detection were two of the key elements that had not been addressed up to that point. And realizing that really diagnostics are really a perfect tool for preventing the spread of disease, right? In the case of an infectious disease pandemic, preventing the spread throughout the population, which then prevents hospitalization rates and a lot of deaths, right? You can obviously also prevent outbreaks and things like that. And we really wanted to create technologies that were really low cost so that anybody could use them, not only people with economic means, but individuals that perhaps live in low resource settings or developing countries. And then we wanted to create something or design something that was very rapid, so that it would enable high frequency testing. Again, this is one of the key elements that really prevents the spread of a disease. If you have something that allows you to test yourself several times a day, let's say, or several times a week, so you can monitor, very frequently your COVID, in this case, COVID status or whatever disease you're interested in. And yeah, so far we've developed 3 prototypes. Two of them are based on electrochemistry technology. the way they work, basically, we can transform the chemical information that is derived from the binding of the virus to a receptor into an electrical signal, and we can pick up that electrical signal very rapidly. We have two different versions. One of them is a benchtop version that you can connect to your laptop, and that one we're thinking more for, at the entrance of buildings and stadiums and schools, etc., to do high frequency testing of whoever goes into a building, so any sort of high density area. And then we have a cell phone version more for personalized use, where you can connect it to your cell phone, and through an app, it tells you whether your saliva sample or nasopharyngeal sample is infected with the virus or not. We just published last week in PNAS. So that first paper was published in Matter from Cell Press. And then just on Friday, a couple of days ago, we published the second prototype in the journal PNAS. And that one, the really interesting thing is that it's made out of graphite, so kind of like the graphite that you have on your pencils, and so it's really low cost and sort of DIY, do-it-yourself DIY, because it's composed of very easy to assemble materials. And that one, we even reduced the cost even more. That one is $1.50 per test. The first one that I mentioned is $4.67 per test. The time of detection is around 5 minutes for all this, between 5 and 6 minutes. And then we've been working on a third prototype that uses colorimetric technology. That's more of a nanotechnology project where sort of like a pregnancy test in the sense that you see a shift in color, but applied to COVID, where when we've basically engineered cotton swabs in a way that when they encounter the virus, they shift color, and so that's how they work. And we've done it. We've demonstrated that with saliva samples and nasopharyngeal samples as well. So yeah, we're excited about the technology development that we've been able to do. It's been a really interesting process for us. Really venturing into diagnostics for viral infections, and we're now applying a lot of these new concepts to other infectious agents as well. So we're expanding to herpes virus. We're thinking about influenza, right? The flu, which every year kills 60,000 people in the US, mostly immunocompromised patients and the older population. So highly prevalent and very important. And we're working on multiplexing as well to not only detect one agent, but multiple agents. And we're doing we're detecting bacterial infections as well. So again, we picture a future where hopefully everybody in their household has some of these tests in a multiplex fashion. So you can test yourself through your saliva or nasopharyngeal samples for multiple infectious agents, perhaps during high flu season. In particular, you can test for flu and other things, including SARS-CoV-2 or any of the emerging variants. And when you travel, you might have a different subset of infectious agents that you want to test for. Let's say if you're in South America or in another part of the world. And so I do envision a future where we have a lot more knowledge about all of that and a lot more ability to test ourselves. But this will really depend on creating technologies that are really low cost, really affordable, so we can have them at home, we can travel with them, and they report back information that is obviously useful to ourselves and empowers us to take next steps, informed steps. If you need to stay at home and quarantine for a little bit, well, that's what you should do. If you're safe, then you can travel or you can do X and Y.
Makes sense. And so, right now, most of the tests are either antibody-based or PCR-based. How does your test kind of stack up against those on the three metrics that seem to matter most? Cost, time to develop, and then accuracy.
Yeah, that's a great question. So I would say in terms of and we've run a number of analysis on this, on the time of detection to cost matrix, our tests are positioned very, very well, because the time of detection, again, is less than 6 minutes for all of them, and the cost is very low. So for the idea mentioned, for the colorimetric test, the cost is 15 cents per test. And so really it's a lot cheaper than anything that is currently out there in the market. In terms of accuracy, the accuracy, I mean, is all reported on the papers, but it's around, I would say, between 85 and 90%. It really varies. So it's not perfect. The tests are not 100% accurate. And then in terms of analytical sensitivity, like the ability to pick up small amounts of the virus, they're actually, the electrochemically based technologies are, they compare very positively with even qPCR. So we can detect really minute amounts of the virus.
Interesting. And so what are the major challenges in developing these diagnostics, and then kind of the next step, making sure they actually get into the hands of patients?
I think the next step really is so to obtain FDA approval for some of them, and to continue to develop them, right? So for some of the prototypes, I think we need to probably improve the shelf life. So we've done a number of shelf life experiments. So they're quite stable, at 4 degrees, but that still needs refrigeration once they are functionalized. So that's one element that could be improved in the future, to extend that shelf life. And then probably automating the whole process of functionalizing the electrodes. We've been doing it manually, mostly manually. I mean, we do have a screen printer, which is kind of like a 3D printer that allows us to print out these electrodes that we use. But automating the whole process and the functionalization process in the future will be essential to increase accuracy, reliability, and all those aspects that we very much care about.
Awesome. No, this is great work, and hopefully this will have an effect on future and pandemics, and maybe even the flu season, or treating or diagnosing antibiotics resistant bacteria. Could be very useful.
I hope so. Yeah, that's our hope.
Yeah. So thank you so much. Your lab is working on some fantastic projects. Just a couple last questions to close this out. The first one is, where can people learn more about your work?
Sure. So we have a website. If you Google, the La Fuente lab at the University of Pennsylvania, you'll find it. I'm also on Twitter, if anybody's interested. It's De La Fuente UPENN. So UPenn. Yeah, I think those are probably the best 2 places where you can interact with me, and also through email, I guess that works as well. It's CFUENTE, cfuente@upenn.edu. Also happy to address any questions or talk to people through there.
Awesome. And before we go, do you have any last shameless plugs or closing thoughts?
No, I just wanted to say, thank you so much for having me. It's really been a pleasure talking to you in this wide-ranging conversation, and keep up the great work with the podcast. And I look forward to listening to the next episodes.
Well, we want to thank you too. This was an absolutely incredible episode, and I had a lot of fun. We're really grateful for your time. So thank you so much, César.
Thank you. Take good care. Bye-bye.